Good morning, everyone, and thank you to the team at Noble for inviting us to present today. Before we begin, I'd like to remind you that statements made during this presentation relating to Eledon's expected future performance or plans may include forward-looking statements that are intended to be subject to safe harbor protection. Please review both this slide for details as well as our risk factors in our publicly filed SEC documents. I'm pleased to present today with Steve Perrin, our President and Chief Scientific Officer, who will be available to answer questions after my comments. We started this year with a new name, Eledon Pharmaceuticals, to highlight the new direction that our company is pursuing. Eledon was formed last September through the concurrent acquisition by Novus Therapeutics of Analyxis Pharmaceuticals, a private immunology-focused company, and the completion of an over $108 million private placement financing to provide our company with sufficient capital to complete up to 4 phase 2 clinical trials with our lead asset, AT-1501. As I'll explain in more depth, we believe that we are developing a potentially best-in-class immune-modulating therapy in AT-1501. AT-1501 is an anti-CD40 ligand antibody. Our strategy for Aladdin is thus to build a company focused on patients for whom anti-CD40 ligand and related therapeutics may provide a potentially life-saving treatment option. As a result, our vision for Aladdin is clear, and we will be focused, we will be agile, and we will be guided by the data and by the science. We selected our indications for AT-1501 based on preclinical data that was generated with either our molecule or historical anti-CD40 ligand molecules. Preclinical data in animal disease models suggest that blocking CD40 ligand is the most efficacious way to prevent acute and long-term transplant rejection in renal and islet cell transplantation, and in some cases can induce long-term transplant tolerance. Blocking CD40 ligand has also been shown to be more effective at preventing allograft transplant rejection compared to blocking the CD40 receptor. The ability to prevent acute and chronic transplant rejection in the absence of calcineurin inhibitors has the potential to transform the clinical management of preventing graft rejection, thus potentially mitigating the nephrotoxicity, diabetes, and increased opportunistic infections often associated with calcineurin inhibitors or CNIs. Islet cell transplantation has the same upside as kidney transplantation, with additional ability to potentially benefit directly from the elimination of CNI's toxicity on islet cells. Chronic exposure to CNIs in organ transplantation is associated with an increase in type 1 diabetes. This is due to the toxicity of CNIs on insulin-producing beta cells in the pancreas. Similarly, CNIs are toxic to transplanted islet cells. Up to 50% of transplanted islet cells may die within days of transplantation, in part due to CNI toxicity, thus exposing subjects to risk of transplant rejection and requiring multiple islet cell transplants in order to obtain a sufficient islet cell mass to eliminate the requirement for exogenous insulin. In autoimmunity, we are focusing on autoimmune nephritis or autoimmune diseases of the kidney. There is a long history of preclinical and clinical data demonstrating that blocking CD40 ligand signaling ameliorates disease progression, modifies biomarkers of disease, and improves renal function in diseases such as focal segmental glomerulosclerosis, or FSGS, lupus nephritis, or IgA nephropathy, for example. Soluble CD40 ligand often correlates with disease flares in autoimmune diseases such as lupus. The clinical program in ALS is based on positive preclinical data in a rodent model of ALS conducted at the ALS Therapy Development Institute, or ALS-TDI. ALS-TDI has screened approximately 500 compounds in rodent models of ALS And according to ALS TDI, blocking CD40 ligand function is one of only a handful that slows disease progression, improves survival, and improves muscle function in these models. Similarly to how soluble CD40 ligand can correlate with flares in autoimmune diseases, soluble CD40 ligand is also correlated with disease progression in ALS. Our pipeline is based on this scientific backbone that I just described. Eledon is enrolling a phase 2 study in ALS, and we are poised to initiate up to 3 additional phase 2 studies this year, including an islet cell transplantation, renal transplantation, and autoimmune nephritis. We expect to begin having data readouts for our pipeline, including interim readouts, starting in the first half of 2022. With regards to timing, I'd like to make a brief comment on the ongoing COVID-19 pandemic. The pandemic has had the unfortunate effect of disrupting drug development timelines around the world, and as such presents an execution risk. Clinical trials across many indications and all stages of development have experienced enrollment challenges and some delays resulting from COVID-related restrictions and impacts. With COVID case counts peaking again globally, and with the emergence of a new viral strain, we are monitoring the developments carefully and are in close contact with both our investigators and our trial sites. For islet cell transplantation, we are doing the trial in Canada and already received clearance from Health Canada to proceed. Because of COVID, however, our study site recently voluntarily and temporarily paused performing islet cell transplants. At this early stage in the trial, we may benefit from a resulting patient backlog to recruit faster, and we therefore believe that we will still be able to generate interim data in islet cell transplant in the first half of 2022. Moreover, depending on when the site starts performing these transplants again, we may ultimately be able to meet our target of a first patient enrolled in this quarter. CD40 has been an attractive drug development target for decades. The engagement of these receptors plays a pivotal role in immune system activation by mediating both antibody and cellular immune response through the modulation of germinal cell function and B cell responses, as well as the polarization of pro-inflammatory T cells. The initiation of a robust immune response is mitigated by co-stimulatory signaling when CD40 ligand binds co-stimulatory receptors, such as CD40, on antigen-presenting cells, as these antigen-presenting cells are simultaneously presenting foreign peptides on their MHC receptors to the T cell receptors on lymphocytes. The interaction of CD40 ligand and CD40 receptor results in clonal expansion, antibody production, and the secretion of pro-inflammatory cytokines that amplify an immune response. Importantly, targeting CD40 is not associated with systemic lymphopenia. Our lead program, AT-1501, is an IgG1 anti-CD40 ligand antibody lacking Fc effector function. It has high affinity binding to CD40 ligand and has shown good manufacturability and good biological activity. It has a half-life of up to 26 days in humans. We are focusing our efforts on the development of an antagonist antibody targeting the ligand rather than the receptor, since targeting the ligand has been shown to be more efficacious in preclinical models of autoimmunity, as well as in the prevention of acute and long-term allograft transplant rejection. This preclinical efficacy may be mediated by several factors, including differences in the cellular distribution of the receptor versus the ligand, since the receptor is present on more cell types than the ligand. Anti-CD40 ligand antibodies also inhibit both CD40 as well as CD11 co-stimulatory receptors on antigen-presenting cells, thus inhibiting the pro-inflammatory polarization of CD4 positive and CD8 positive lymphocytes. In other words, targeting anti-CD40 ligand may be a $2 for the price of one opportunity. Finally, blocking the ligand also polarizes CD4 positive lymphocytes to FOXP3 positive Tregs, which functionally secrete IL-10 and other cytokines, creating a tolerogenic environment in the context of preventing allograft transplant rejection and autoimmunity. To date, we have completed a phase 1 single ascending dose study of AT-1501 in both healthy volunteers and people living with ALS. In this study, AT-1501 showed a good safety profile with low ADA responses that were not dose-related. In the 8 milligram per kilogram cohort of the phase 1 study, we conducted an immune challenge with KLH. KLH is a shellfish protein that was injected under the skin or subcutaneously. In the control subject on the far right that you see on the slide, there is a robust antibody response to the foreign KLH protein, which peaks at 15 days. AT-1501, however, completely abrogated an immune response to KLH in treated subjects 1 and 2 on the left, as well as partially inhibited the immune response in subject 3. I'll now run through our indications and related data in more detail. There are an estimated 23,000 renal transplants performed yearly in the United States, and about 200,000 Americans are living with a transplanted kidney. Still, there remains a 3 to 5 year waiting list with over 90,000 people, and yet up to 15% of all of the kidney transplants in the United States every year are retransplants due to graft failure. If grafts lasted longer, more kidneys would be available for new patients every year, and waiting times would go down. CNIs are critical components of immunosuppressive regimens to prevent acute and long-term transplant rejection. However, chronic exposure to certain CNIs, including tacrolimus, is associated with nephrotoxicity, an increase in opportunistic infections, increased malignancies, and an increase in type 1 diabetes due to pancreatic beta cell toxicity. These liabilities may result in a requirement for reduced exposure to CNIs over long periods of time, and as such, a decrease in the ability to prevent long-term rejection. We believe that AT-1501, in combination with elimination of CNIs from renal transplant procedures, may increase long-term graft survival and significantly eliminate the toxicities associated with long-term exposures to CNIs. Several historical studies have described the effects of anti-CD40 ligand antibodies in animal models of renal transplantation. These have shown that anti-CD40 ligand therapy can prevent both acute rejection and long-term rejection in non-human primates. In one of these models, the lack of immunosuppressive treatment results in acute transplant rejection in only about 14 days. Chronic administration of anti-CD40 ligand antibodies as a monotherapy for 5 months, however, inhibits transplant rejection with good kidney function as measured by proteinuria. Another historical study examining an anti-CD40 ligand therapy in combination with current immunosuppressive regimens resulted in a mean survival of greater than 300 days in animals in the combination group. Moving over to islet cell transplant, there is currently a limited number of islet cell transplants occurring in the United States. Yet there are over 1 million people in the US with type 1 diabetes, of whom an estimated 70,000 live with the so-called brittle phase of the disease, where they can experience extreme swings of blood glucose levels, glycemic unawareness, where they don't know their blood sugar levels, and be at risk for significant comorbidities and even death. Adoption of islet cell transplant as a treatment option for brittle diabetes has been hampered in part by the toxicity associated with the CNIs that are required to prevent islet cell rejection, yet result in acute islet cell loss and dysfunction. CNI toxicity and islet cell loss often results in requirements for multiple islet cell transplant procedures in order to eliminate the necessity for exogenous insulin and to have optimal glucose control. The toxicities associated with CNIs also increase the risk of severe renal dysfunction in a patient population where renal function may be already compromised or at risk. The elimination of CNIs in islet cell transplant could thus potentially transform treatment options for people living with brittle diabetes. Historical studies in non-human primate models of islet cell transplantation showed that treatment with anti-CD4 ligand antibodies induced long-term islet cell function and graft survival, even as monotherapy in the absence of additional immunosuppressive agents. We have expanded on these early observations with AT-1501 in a non-human primate model of islet cell transplant, comparing the safety and efficacy of AT-1501 with an immunosuppressive regimen containing CNIs. Animals treated with AT-1501 had better graft function, improved appetite, and significant weight gain a year after transplant, compared to standard immunosuppressive regimens, where acute rejection was observed more frequently, or in the case of the animal in this slide, he experienced kidney failure and needed to be euthanized. Animals undergoing islet cell transplantation with AT-1501 monotherapy had significantly higher C-peptide levels immediately after transplant. The islets with AT-1501 monotherapy appeared healthier and could dynamically sense glucose levels, as demonstrated by functional increase in C-peptide levels after eating. We are considering different options for a potential fourth indication in an autoimmune disease of the kidney, but I'd like to highlight these type of diseases by talking about two. Systemic lupus erythematosus, or SLE, is one of the largest autoimmune populations globally, and up to 40% of people with SLE develop lupus nephritis, or lupus of the kidney, and may experience kidney dysfunction, dialysis, and end-stage renal disease. Similarly, FSGS is a rare disease, autoimmune nephritis, that can progress to end-stage renal failure. Proteinuria is often utilized as a biomarker of kidney function and can be leveraged for patient stratification, as well as a surrogate marker for improvement in kidney function. There are currently no FDA-approved treatments for FSGS and only one for lupus nephritis. IgA nephropathy is another autoimmune disease in this category. Systemic steroids and immunosuppressants such as CNIs are widely prescribed. There is a long history of modulating CD40 ligand activity in preclinical models of lupus nephritis and glomerulosclerosis with anti-CD40 ligand antibodies. Anti-CD40 ligand antibodies have shown amelioration of disease progression and improved survival in rodent genetic models of SLE. They have also demonstrated improvement of fibrosis in the kidney and the spleen, improvements in kidney function, and reduction of immune cell infiltrates into the kidney. Systemic biomarkers of lupus, such as anti-dsDNA antibodies, are also reduced with anti-CD40 ligand treatment. Similar data has been described in preclinical models of FSGS. In those models, anti-CD40 ligand treatment decreased immune cell infiltrate into the glomeruli and ameliorated kidney function as measured by a reduction in proteinuria. Moving to our fourth indication, ALS, the role of autoimmunity in ALS has been elucidated and is increasingly recognized. ALS is an orphan progressive neurodegenerative disease with approximately 5,000 new cases diagnosed in the United States every year and a prevalence of 30,000 cases in the United States overall. The average age of diagnosis is only 55 years old, and only about 10% of cases are believed to be hereditary. Despite 2 approved drugs, the median survival remains only 3 to 5 years after symptom onset. The ALS Therapy Development Institute discovered the activation of the co-stimulatory pathway and activation of CD40 ligand signaling at symptom onset in skeletal muscle, brain, and sciatic nerve in a rodent model of ALS. Using a murine antibody that blocks CD40 ligand function in mice, they further demonstrated that blocking CD40 ligand signaling improved muscle function, slowed disease progression, and improved survival. These clinical manifestations are believed to be due to reduced immune cell infiltration of macrophages into skeletal muscle, and they're destroying denervated nerves. The plasticity of the nervous system to repair itself in the absence of this immune cell attack results in improved neuromuscular junction occupancy and improved muscle function. Blocking CD40 ligand signaling also prevents pro-inflammatory polarization of lymphocytes, reduced neuroinflammation, and improved motor neuron survival in rodent ALS models. Eledon initiated a phase 2 study of AT-1501 in people living with ALS in October of 2020. The trial is a 12-week open-label dose-escalating safety and biomarker study with multiple doses of AT-1501. The endpoints of the study are safety and tolerability, changes in pro-inflammatory biomarkers, as well as neurofilament light chain. Exploratory clinical endpoints will also be assessed. I'd like to finish my presentation by reiterating our vision for Aladdin, of focusing on patients for whom anti-CD40 ligand therapeutics may provide a potentially life-saving treatment option. We believe that the biology around CD40 and CD40 ligand is well validated, that CD40 ligand is the optimal target, and that AT-1501 has the potential to be a best-in-class molecule. Financially, thanks to the runway provided by our recent financing, we expect that we have sufficient capital to execute up to 4 phase 2 clinical trials. Finally, we expect to report on our execution in 2021, be able to begin generating data in the first half of 2022, and have multiple interim and final data readouts continuing into 2023. Thank you very much for your time and interest today. As I mentioned, Steve Perrin, our President and Chief Scientific Officer, is present with me as well, and we both look forward to answering your questions. Nate, I think you're muted.
Went back on. Thank you, DA. Thank you, Steve, for joining us today and presenting at the Noble Conference. Everybody's excited about Eledon here at Noble, and we congratulate you on all the progress that you guys have had up to this point, and a very exciting time for a biotech company like yours. So we'll go to some of the questions. The company recently pressed the allowance of the start of an islet cell transplant trial. Can you go through what the significance of that trial will be as far as the data points you're looking to achieve?
Sure. Steve, are you on? Do you want to talk about the trial?
Yeah, sure. Can you hear me, DA?
I can. Good morning.
Sure, thank you. Yeah, so it's a really exciting trial. As DA mentioned, we recently got regulatory approval from Health Canada to start that study. Really exciting study because of the ability to quickly assess mechanism of action of the drug and really see if blocking CD40 ligand signaling can facilitate graft function, improve graft function over what has historically been done with standard of care containing calcineurin inhibitors, where we're really optimistic based on preclinical data that graft function should really be superior in the absence of CNIs. Because of the fact that the islet cells are transplanted into the portal vein of the liver, they'll interact there dynamically and be able to sense glucose signaling. And it really opens the opportunity to look at multiple biomarkers subject by subject to see how their graft function is doing with surrogates of insulin production called C-peptide levels. You can also look at their glycemic awareness events over time, and the ability to really limit or completely get off exogenous insulin. So a really exciting study that we'll be launching here hopefully very soon.
Thank you, Steve. As far as the data points that can come out of the trial, how quickly can that happen with respect to after transplant?
I mean, like I said, it's a very dynamic process. These folks have been on exogenous insulin for very long periods of time with their diabetes. Post-transplant, your goal is to kind of taper them off exogenous insulin within 20 to 30 days, and then you start following their ability of their graft function to sense glucose levels and dynamically respond with the islet cell mass that was transplanted. Typically, they're evaluated by around day 75 to 90 to see how their graft function is doing and to see how all those metrics look. And so it's a very dynamic process. You're talking 2 to 3 months post-graft function to get a pretty good assessment historically on what graft function looks like. That's the time period by which their surgeon would assess whether they need a second islet cell transplant if the first graft is not functioning well enough.
So most patients that get islet cell transplants today require at least a second graft, and often that's known within 75 to 90 days after the first transplantation. Now, our study is going to be open label, but we won't be reporting on patients one at a time. So we'll be reporting on our first set or cohort of patients in the first half of next year.
How many patients will be enrolled in the islet cell?
So it'll be most likely about 6, but we can do up to 12.
And a follow-up question to that, what would you show as a success rate as being positive? 50% success, or would have been the marks that you guys are looking for as far as engraftment?
I mean, I think again we're trying to achieve success over historical numbers here. There's been several well-done studies with Shapiro's lab up in Canada, often called the Edmonton Protocol. There was a really exciting results from phase 2B studies with a consortium of sites in North America that was published a few years ago. And even though graft function there looked pretty good, over 60% of the subjects needed a second transplant, and a lot of that is attributed to the use of CNIs and tacrolimus to prevent graft rejection. So our success here is the number of patients that don't require a second graft and how good their graft function looks like with that initial cell mass at day 75. But the ultimate goal here is to see how we perform in the absence of calcineurin inhibitors.
For other non-human primate trials that have been completed, have other antibodies shown this same type of data with such great success in a non-human primate trial?
So there have. I mean, Biogen's original 5C8 antibody that was used very widely in the scientific community back in the late 90s has been tested in multiple non-human primate models of islet cell transplant and showed very good activity, much like we have even as a monotherapy. So we replicated that data and we're optimizing the dosing protocols and whatnot in preclinical models now. But yes, I mean, other antibodies have shown that you can get a good stable graft function for long periods of time, often over a year, even with monotherapy in the absence of other immunosuppressant drugs in these types of preclinical models.
What data? The current molecules that are in clinical development in phase 2 versus some historical therapeutics, including the one that Biogen had, which had been associated with the historical issues, and so it's no longer in development. We believe we are the only ones that have shown the data the way we have.
Excellent. Thank you. What data will you report for the phase 2 ALS trial, and then how is enrollment going, and how many patients will you enroll in the ALS trial?
Steve, do you want to?
Yes, so the ALS trial, as DA indicated, started enrolling last year. COVID-19 has had a modest impact on that as we've gotten sites up and running. The enrollment's a little bit behind, but not really significantly behind what we thought would be today. So far, several patients have gotten multiple doses of drug. The ultimate endpoint of the trial, as you can imagine, it's a multiple ascending dose study in up to 54 subjects. Safety, tolerability, collecting more pharmacokinetics on the drug is really important in this first multiple ascending dose study in humans. And then obviously, because it's a multiple ascending dose study, it's a biomarker study where we can gather lots of information about the activity of our drug in the context of an autoimmune indication. So we're going to look for target engagement to show that we're actually hitting the CD40 ligand target on lymphocytes and blocking its function. We can look at markers of inflammation in blood that's been well published and well described across multiple publications in ALS over the years. And kind of the other biomarker program that's a part of this, even though it's a fairly short study, it's only a 12-week study, we are going to look at neurofilament light chains in circulation. There's been much excitement in the field that we might finally have a biomarker of neurodegeneration. When nerves get sick, both in ALS, Alzheimer's, and other neurodegenerative diseases, the dying nerves, often as they're being apoptosed and degraded, neurofilament light chain, which is a structural protein, ends up in the cerebral spinal fluid, which ultimately bathes with your circulation so you can measure neurofilament light chain in blood. So we're going to be looking at neurofilament light chain levels to see if we change that over the course of the study. But again, it's a fairly short study, so we may not see that one.
Yeah, so we have multiple sites up, multiple patients, with patients having received multiple doses. It's proceeding. The trial continues to proceed as planned, and we expect to have data in the first half of next year as planned.
Thank you. Can you talk about the competitive landscape there in ALS and what that looks like, how you guys feel you're positioned with your phase 2 asset?
I mean, there's 2 FDA approved drugs for ALS, as we know. Relutrec was approved over 25 years ago. And more recently in 2017, Darafone was approved for ALS. Relutrec's been well adopted into the community pretty much globally at this point. Darafone, still it's having modest effects at best, but neither one of these drugs is truly disease modifying. They're not really slowing down disease to the point that they're changing time to event, changing survival kinetics, improving quality of life. So there's still a significant unmet need, even with 2 FDA-approved drugs on the market today for ALS.
Another question for islet cell transplant. Are there any other companies doing islet transplant studies that you know of?
DA, do you want to take this one?
Yeah, Steve, happy to let you do it.
I mean, there are other companies in the space trying to develop technologies that could facilitate transplanting islets into people with type 1 diabetes. ViaCyte comes to mind as a company that has a technology platform that is trying to engineer cells with evading the immune system to get cells into the body that can sense glucose levels and secrete dynamically insulin to respond to glucose levels. Semma was developing stem cell lines, as we know, and was acquired by Vertex a little over a year ago, also developing a technology platform from stem cells that are terminally differentiated, can secrete insulin dynamically in response to glucose. And again, I think the challenge with the other companies that are in the space today is their technology platform sufficiently developed to evade the immune system's response to wanting to attack those transplanted cells, or are they going to need some type of immunosuppressant technology or cocktail that we've used historically to inhibit the immune system from attacking those cells and eliminating them.
And we got time for just a few more questions. What is the cost of the Eyelid cell trial and the ALS trial?
So we have not provided detailed cost breakdowns of our trials, but what we have said is we have sufficient capital to finish all up to 4 trials, so all of our trials.
Okay. And I think we'll probably have time just for this last question. How many sites will the kidney transplant trial enroll? And can you give us a sense of what that trial looks like and what endpoints you'll be looking for there?
So we haven't given specific details on the trial or trial design, and we'll do that as we get closer to getting to launching the trial. But Steve, do you want to give high-level details on the endpoints?
Yeah, I mean, clearly there's a lot of FDA guidance on what the endpoints of a renal transplant study should look like. So, minimally you have to get out to 12 months and show some type of non-inferiority to standard of care, which is, again, typically an immunosuppressive cocktail containing both induction therapy as well as long-term maintenance therapy. And you have to hit metrics on graft function, patient survival. It's a very well-defined FDA guideline on a triple endpoint at 12 months that people tend to follow.
Is that going to be a difficult trial to enroll as far as kidney transplant goes? How many patients are available? And
I mean, it's a very active space as far as technologies, because everyone recognizes the significant unmet need of the immunosuppressant cocktails that we're using today. They're very effective at inhibiting acute graft rejection, but they have longer term liabilities with associated toxicities that are very hard to maneuver around for subjects, and the tolerance of those drugs long term are very, very challenging. So we think it's a very good opportunity for us. It's a fairly large market, and this could be really transformational in the context of eliminating calcineurin inhibitors from the standard immunosuppressive maintenance cocktail.
okay, one last follow-up question. As far as safety goes with your drug, how have things played out up to this point, being that safety is an important factor in all of the trials you're going to be doing?
I mean, we launched into testing in humans after very, very extensive safety assessments in preclinical non-human primate models, looking very carefully at platelet activation and evidence of thromboembolic activities, and haven't seen any of those in our preclinical tox packages where, we dosed very frequently 6 months with very, very high doses. Our phase 1 study in primarily healthy volunteers also showed a very good safety signal. Again, looking very carefully for evidence of platelet activation, and haven't seen any. There was more serious adverse events in the placebo arm of our phase 1 study than there was in the treatment arm. I mean, obviously, we're in our first multiple ascending dose study in ALS, and it's really exciting that we're going to be building on that safety database from the phase 1. And we'll continue to build that safety database across all 4 of our phase 2 studies, which is really exciting.
DA and Steve, thank you very much. We're out of time, but we really appreciate the presentation, and congratulations again on all the progress.
Thank you. Good to be here.